Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And what we were discussing about the different aspects of the cell and molecular biology in this particular course. So far we have discussed about the cell biology, we have discussed about the cell division and the cell nutrition, cell uptake, and many aspects related to cell signaling. And then subsequent to that, we have also discussed about the central dogma of molecular biology. So where we have discussed about the replication, transcription, and translation. And subsequent to that, we have also discussed about the various aspects related to the genetic material of the organisms. We have also discussed about the different characters, how the characters are being inherited between the different generations and so on. So these were some of the very important aspects of the the cell and molecular biology. Continuing to that, we are also going to discuss today about the immunology, right? And we are also going to discuss how the immune system is protecting and how the immune system is playing a crucial role in the survival of an organism. So when we talk about the immune system is about the system which actually going to resist for the growth of the external organism. Remember that if the organisms are surviving, they are surviving because they are being protecting themselves from the any kind of threats from outside and as well as from the inside as well, right? So these external threats are mostly been coming from the other organisms, and there are internal threats which are also coming because of the some of the metabolically induced antigens or some of the internal problems which are going to be encountered by an organism. So the main focus of the immune system is that it should resist against the changes which are being going to be induced by the external or the internal sources. So when we talk about the study of the immune system is being done by a branch of science which is being called as the immunology, right? And immunology is the study of the immune system, and as we said, the immune system is a system which will be present in all the organisms, whether it is a prokaryotic in origin or the eukaryotic in origin. Although in this particular module, we are not discussing about the immune system present in the bacterial system. We are only focusing on the immune system what is present in the eukaryotic system, especially the multicellular eukaryotic system like the humus. So the immunology is the study of immune system, the body's sophisticated defense network that protect against the pathogens such as bacteria, viruses, fungi, and parasites, and as well as the harmful substances, right? So when you will discuss about the different aspect, you will understand that it is not that the external organisms are only being the threats for the human body. It is also the internal harmful substances which are being produced by many mechanisms, and many of these substances are also being taken up or being taken care by the immune system. So as a subfield of biology, the immune system or immunology explore how this system evolved to defend the multicellular organisms for a wide array of microscopic and as well as the macroscopic invaders. Now talking about the what is the scope of the immune system. So the scope of the immune system is the immune system is highly adaptable, right? So that is one of the important feature of the immune system, able to respond to the threats as small as the 30 nanometer poliovirus and as large as at the 100 centimeter long parasitic worms like the Dictyostelium rhinale, right? So you see that the immune system is very, very diversified and adaptable. It is actually going to be respond to as small as a tiny particle of a virus, or it could be responding to a large parasitic worms, right? To handle such diversity, it uses a broad array of recognition and destruction mechanisms supported by a dynamic network of the cells, molecules, organs, and the pathway. So currently, in this particular lecture, we are only going to discuss about the basics of the immunology, and we are also going to discuss the different components what are being important for the immune system to execute their the immunological responses. So in that we need to discuss about the recognition and destruction mechanisms. We have to discuss about the cells, molecules, organs, and the different types of pathways. Because of this complexity, the immune system engage in a coordinated and sometimes the circular process, making it one of the most integrated biological system to study. To play the critical role in many groundbreaking medical achievements that that will be responsible for the eradication of the smallpox via the vaccinations. I'm sure you might have studied or you might have heard about the many stories about how the smallpox is being eradicated by the vaccinations and so on, and that's how the people understood the concept of the immune system and the concept of the defense mechanisms. Then there are so many examples of the organ transplant, right? So, and then there will be a treatment for the autoimmune disorders, allergies, and the asthma, right? So many of these terms could be very complicated for you. You may not be able to understand these, but once we discuss about these terms in more detail, you will be able to understand what is mean by the autoimmune diseases, allergies, and asthma. Now the question comes, why would like to study the immunology, right? So the understanding the immunology is a crucial in today's world. Why? It is because every day there will be a new infection disease arises, right? You might have heard about the coronavirus or the COVID-19. There are so many times you might have heard about the many new viruses are being discovered, and they are also causing the different types of new diseases. Then the existing diseases, the organisms are actually acquiring the antibiotic resistance. And then you might have already experienced what will be the impact of the pandemics, right? So we have already seen the impact of the pandemic when we seen the impact of the COVID-19s. And then there is an urgent need to explore how the immune system works and how it can be supported or the modified, right? So remember that when people were, getting the severely affected by the COVID-19 or the coronavirus, the immune system was playing a crucial role, and because we know that how the immune system is handling this particular virus, we devised or we advised the people to take different types of medicines and different types of modulators, right? So basically, immunology provides the insight into many things, right? It gives the insight about how the disease is spread and how it can be prevented. It gives you the insight about why some immune responses fails or go overboard, right? For example, the development of the autoimmunity or the allergies. And then it also discusses about how to enhance the immune responses through the vaccination and as well as the different types of immunotherapies. What is the advantage of studying the immunology? So because the immunology is going to give you a tool to or a weapon to work against a particular infectious organisms or particular type of foreign substances, you can actually be able to use this particular knowledge to develop the different types of molecules, right? So studying the immunology offers the numerous practical and scientific benefits. For example, it will help you in terms of developing the vaccines, right? So there are different types of vaccines available which are actually helping us to protect against the different types of viruses. So basically the vaccine development, it is actually trained the immune system to fight against the infectious organisms, right? Then number two, immunotherapies, right? So it harness the immune system to fight the cancers and the chronic diseases. Number three, transplant success, right? Because we know that how the immune system is going to respond against the different types of the foreign the organs from the different donors. We actually can use that information to help to prevent the graft rejections and the how the you can actually be able to bring the compatibility between the two individuals so that the transplant could be successful. The number four is the integrated health, right? So it reveals the connection between the immune system and the nervous system, endocrine system, metabolic system, and helping to maintain the homeostasis for the overall well-being of a person, right? Now, if you talk about the development of the immunology as a field, it started a way back in the 430 BC, when the people have realized that the existence of the plague in the era of the Athens, right? And then in the 15th century, Chinese and Turks actually suggested that the dry crust derived from the smallpox pustules were either inhaled into the nostril or inserted into the small cut of the skin, and that actually is going to give you a protection against a disease. Then in the year of 1978, the method of variolation was improved by the Edward Jenner, and that's how he actually discovered a vaccine against the smallpox. Then in the year of 1880 and 1881, the Louis Pasteur developed the vaccine against the cholera that is causing the organisms, right? So you remember that cholera is being caused by a bacteria which is called as Vibrio cholera, right? And the Louis Pasteur actually discovered a vaccine against the cholera. Then 1890, the Boehringer and the Shivapuro gave the first insight into the mechanism of immunity, earning the Nobel Prize in the medicine in the year of 1901. Then in the year of 1930, the Elvin showed a fraction of serum called gamma globulin, and that is responsible for the antibody activity and the immune responses. Then in the year of 1940, the Merlin Chase succeed in transferring the immunity against the tuberculosis organism by transferring white blood cell between the guinea pigs, right? And the year of 1950, the scientists identified a cell which is called as lymphocytes, contributed both into the humoral as well as the cell-mediated response. Then the year of 1956, the Bruce actually at the Mississippi State University indicated there were two types of lymphocytes. You have the T lymphocytes and then you have a B lymphocytes, and the T lymphocyte is involved into the cellular immunity, whereas the B lymphocyte is involved into the humoral immunity. And you will understand about what is meant by the cellular immunity and the humoral immunity in a due course of time, and we will discuss about these terms. Then the year of 1966 and 67, the scientists discovered the immunoglobulin E and its role into the allergic reactions and the allergic research, right? So then in the year of 1975, the Kohler and Mistine developed the hybridoma technology, and that's how he has started developing the monoclonal antibody, which become essential tool in diagnostic and the therapeutics. The year of 1987, the Tonigawa received the Nobel Prize for elucidating the genetic mechanism behind the antibody diversity, explaining how a vast variety of antibodies can be produced from a limited number of genes. And this is a very, very important mechanism, very, very important discovery, which actually explains how a single gene could actually give rise to a different types of antibodies. Then the year of 2005, the Fraser developed the first vaccine targeting the human papillomavirus or HPV, and he rolled the he bring the correlation between the HPV infection and the incidence of the cervical cancer, and that's how we still actually give a vaccine against the HPV infections, and that protects the person from getting the cervical cancer. Then the year of 2010, the FDA approval of the first immune checkpoint inhibitors for the metastatic melanoma, and then the post 2010 till date, there are so many discoveries. People have developed the messenger RNA vaccines. People have developed the the messenger RNA vaccines like different types of personalized vaccines and so on. So immunology is a very, very important field, and it is developing every day actually, and it is making newer and newer discoveries. First, what we are going to understand is we are going to understand about the immune system, how the immune system is functioning against a particular organisms or particular infectious diseases, what are different components of the immune system, and then we will also going to take up the some aspects related to the immune system. So, what is the basics of the immune system? So, vertebrates are protected by the two different types of immune systems. We have the innate immune system and the adaptive immune system. Innate immune system, which you are going to get from your birth, right? So, this is the system what you are going to get from the birth, and adaptive immunity is the immunity what you will acquire, while you are going through with the process of the birth to the adulthood. So when you go from the birth to adulthood, you are actually going to develop the adaptive immunity or the acquired immunity. So these two sides have collaborate to protect the body against the foreign invaders. So this is actually the innate immunity is actually the starting material, right? You are actually going to bond with the basal level of immunity against the different types of organisms. But that basal level of immunity can be trained over the course of time when you are going to grow from the small baby to to adult, and that's how you are actually going to train your system so that you are going to develop adaptive immunity, right? So what is the innate immunity? Innate immunity include the built-in molecular and cellular mechanisms that are encoded into the germline and are evolutionary more primitive, aimed at preventing infection or quickly eliminating the common invader. So innate immunity is actually a immunity which actually going to protect you, as soon as you are taking the birth, right? So that time there will be infectious organism which will start attacking on you, right? So they will be actually get protected from the different types of these organisms by the help of the innate immunity, right? A second line of immunity, which is called as the adaptive immunity or the acquired immunity, is much more attuned to the subtle molecular differences, and this part of the system which relies on the B and T cells taking longer to come on board, but it takes much more antigenic specific. Typically, there is an adaptive immune response against a pathogen within 5 to 6 days after the barrier breach and the initial exposure, followed by a gradual resolution of the infection. So basically, when an organism is entering into the system, it is the innate immunity which is actually going to protect this, right? And it is actually going to take up that, and then it is actually going to start producing the immune response, right? And that immune response would be adaptive immune response, right? So for understanding the immune system, we have to first understand about the innate immunity, and then we also have to understand about the adaptive immunity. So what is the innate immunity? As I said, innate immunity is the what you are going to born with, right? So for example, when you are born with two legs, two hands, and so on, right? So these are the basal material, right? But after that, you use these hands, you use these legs, and that's how some people are, becoming a wrestler, some people are becoming the runners, some people are becoming the cricketers, some people are, so that is their adaptive immunity, right? Or that is the adaptive features, right? So by birth, we all are born like similar, right? We all are having the two legs, two hands, and so on. But during the course of your development, when you are a small kid to adult, you develop some of your skills, right? And that's how it becomes a different type of person, right? So that is what the difference between the innate immunity versus the adaptive immunity. So innate immunity system includes the anatomical barriers against the infection, such as skin, both physical and the chemical, and as well as the cellular responses. So the physical barriers, the body's first line of defense, are the epithelial layer of the skin and the mucosal and the granular tissue surfaces connected to the body's opening. These epithelial barrier prevent the infection by the blocking pathogen from entering into the body. So within the immunity, you have the physical barrier, and you also going to have the chemical barrier, right? So these are the first line of defense, like, which means like you are going to have the two different types of doors. You are going to have the door to protect the person for entering into your home, right? So you can have the physical door, you can also have the chemical door, which means you can actually have some kind of spray or something, right? Then you have the chemical barrier. So at these surfaces, you have a specialized soluble substance that processes the antimicrobial activity, such as acid, right? For example, in our gut, you are going to have a large quantity of acid, and that acid will not allow any organism to get into the body. Then pathogens that breach the physical and chemical barrier due to the damage or to a direct infection to the epithelial lining can survive into the extracellular space, or they can infect the cells, eventually replacing and processing the spread to the other part of the body, right? So, what are the anatomical barriers of the infection? So, the most obvious component of the immune system are the external barrier to the microbe invasion, the epithelial layer that insulates the body's interior from the pathogen of the exterior world. These epithelial barrier include the skin and the tissue surfaces connected to the body's opening, the mucous epithelial lining like the respiratory, gastrointestinal and urogenital tract, and the ducts of the secretory glands such as the salivary gland, lacrimal gland, and the mammary glands, which produce the saliva, tears, and the milk respectively. Skin and other epithelia provide a king of like plastic wrap that encase and protect the inner domain of the body from infection. But these anatomical barriers are more than just a passive wrapper. So they contribute to the physical and mechanical process that helps the body to shed the pathogens and also generate the active chemicals and biochemical defense by synthesizing and deploying the molecules including the peptides and protein and that have a into microbial activity. So anatomical barriers are actually going to resist against the entry of that pathogen, right? Once they enter, then the active molecules are actually going to fight against these particular in foreign particles. So these are the some of the places where you are actually going to have the protective, like for example, you are going to have a skin. So the skin is going to have the antimicrobial peptide and fatty acid in the sebum, right? So like the then you have the mouth and the upper alimentary canal, right? So you have a mouth, right? So there you are going to have an enzyme, the antimicrobial peptides, and the sweeping of surface by the direct flow of the fluid toward the stomach, right? So within the enzyme, for example, you have the lysozyme, right? So your saliva contains the large quantity of lysozyme, and lysozyme is actually killing the bacteria, right? So uh, that is something what you are supposed to do, right? Then within the stomach you are going to have a low pH because the the stomach is having the low pH because of the enzyme which is called SCL. Then it also has the digestive enzyme. Then you have an antimicrobial peptides and the fluid towards the intestine. So it is actually having the running fluid also. So because of that, it will not allow any molecule to remain. Then within the small intestine, you have a digestive enzyme, antimicrobial peptide, and fluid to the large intestine. Then we have a large intestine, the normal intestine flora compete with the invading microbes, and then you also have the fluid or feces expelled from the rectum. Then you have airway and the lungs, right? So these are the airway, right? From the nostril, the airway go all the way towards the lungs, right? So you have the cilia which sweep the mucus outwards. You have a coughing, sneezing, and then you also have macrophages into the alveoli of the lungs, right? So remember that you are always having the running nose when there will be an infection, right? So that running nose is because you are actually expelling the mucus, right, from your nostrils. And then the nostril is also having the hairy surfaces, so that also protects you from the all the organisms which are coming through the dust particles. Then we also have the urogenital tract. So it is flushing by the urines, aggregation by the urine mucins, then low pH, antimicrobial peptides, proteins into the vaginal secretions. Then we have the salivary, lacrimals, and the mammary glands, right? So all of these glands are actually secreting large of fluids, and the flushing by the secretions and the antimicrobial peptides and the proteins into the vaginal secretions. And then we talk about the adaptive immunity. So adaptive immunity is actually going to take up the material what is being present within the innate immunity. Then it is actually going to train that system. So adaptive immunity is nothing but it is actually going to train your innate immunity, and that's how it is actually going to allow them or give them a training to fight against the different types of infectious organisms. So one popular, significant and unique attribute of the adaptive arm of the immune response is the immunology memory, right? So you actually brings the the immunity, right? And you actually keep a memory of that immunity. So for example, if there will be a memory against the Mycobacterium tuberculosis, right? As even suppose I got the infection now and I got the memory 10 years back, the moment I will get the infection, the system will remember that, okay, how actually I will going to kill this particular organism, because it has a memory, right? So it will actually going to start producing the different those kind of molecules which are actually going to taken care of these organisms. So this is the ability of immune system to respond much more swiftly and with a greater efficiency during a second exposure to the same pathogen. Unlike almost all other biological systems, the vertebrate immune response also evolved not only the ability to learn to encounter with the foreign antigen in real time, but also the ability to store this information for the future roles. And this is a very important feature of the adaptive immune system, that it not only fights with the current invaders, it also keeps a memory that in case the similar kind of invaders are going to come into the body, I will actually not going to go through with all the procedures of preparation, but I will actually going to know what I supposed to do. During the first encounter with the foreign antigen, the adaptive immunity undergoes what is termed a primary response, and during which the key lymphocyte that will be used to eradicate the pathogens are clonally selected and utilized to resolve the infection. These cells incorporate messages received from the innate players into a tailored response to the specific pathogens. Now, all the subsequent encounter with the same pathogen are referred to as the secondary response, right? So, in the primary response, the body will understand how to tackle this particular problem. It will actually going to generate the memory. So, which means actually it is actually going to generate a protocol how to encounter any such pathogen when it comes to the second time. But when it comes to the second time, then it's actually going to generate the secondary response, which means it is not going to go through the whole process. Instead, it will actually going to produce that response so that this particular pathogen should be removed. So during a second pathogen, memory cells, which are actually going to be having a task of bringing that protocol back, basically going to produce the B and T lymphocytes, which are already been trained to produce the primary response, and then it is actually going to be put it into the fight, so that it is actually going to produce the antimicrobial molecules, and that's how it is actually going to eradicate that particular pathogen. Those cells begin almost immediately and pick up the fight where they left off, continue to learn and improve their eradication strategy during each subsequent encounter with the same pathogen, right? So this is what we have said, right? So if you have the memory, you will know how to kill the person, right? For example, if two wrestlers are fighting, right? So when the first time they will be encountering, they know they will actually going to use the different types of tactics to to fight and defeat the person. But when the same encounter happens the next time, then it will not go through with all the tactics. It will know the final tactics, how it has been defeated the person, and he will use that tactics, and that's how it is actually going to taken care. So the memory lymphocytes provide the mean for subsequent response that are so rapid, antigen specific and effective that when the pathogen infect a body second time, dispatch of the offending organism occurs without any symptoms. And this is actually the purpose when you are doing a vaccination, right? You are doing a vaccination basically to produce memory lymphocyte, right? So that when you are going to do a vaccination, you are mimicking the entry of that particular pathogen into the body, and so that you can actually be able to generate the memory response, and this memory response will keep the whole protocol ready. So as soon as the real pathogen will enter, it is actually going to taken care of that particular infection. It is a remarkable property of memory that prevents us from catching many diseases by the second time. So immunological memory harbored by residual B cell and the T cell and is the foundation for the vaccinations, which uses the crippled or the killed pathogen as a safe way to educate the immune system to prepare it for the later attack by the life-threatening pathogens. Now, what is the difference between the innate and the adaptive immunity? So, innate immunity, in terms of response time, the innate immunity takes the minutes to hour, whereas the adaptive immunity goes into the days, right? Because innate immunity instantly works, and that is the purpose of having an innate immunity. As soon as the pathogen enters, you are supposed to have some resistance mechanism so that you can actually be able to stop its entry for some time, right? And during this time period only, the person is going to evolve the adaptive immunity, and that's how it is actually going to be able to defeat the immune response. Then specificity, the specificities are limited and the fixed. So it is actually having a gross response rather than a specified response. Whereas in the adaptive immunity, it is highly diverse, adapts to the improved during the course of immune response. Then response to the repeated infection. So innate immunity responded every time at the same time, right? Same way actually. Whereas the adaptive immunity becomes more rapid and effective with each subsequent exposure. Then you have a major component, so you are going to have a barrier, for example, the skins, and then you have the different types of cells like phagocytes and pattern recognition molecules and so on. Whereas in the adaptive immunity, you have the lymphocytes like T cell and B cells, and then you also have the antigen specific receptors and antibodies. Now, within the innate immunity and the adaptive immunity, you probably could have the two different types of systems. You can actually have the system where you are actually using the liquid component, or you can also have a system where you are actually going to use the cells. So, if you use the liquid component, then it is going to be called as humoral response or humoral system. If you use the cells, then it is actually going to call the cell mediated system, okay, or cell mediated response. So let's first discuss about the humoral response. So humoral system, so humoral immune system, also known as the antibody mediated immunity. Basically, humoral immunity response means all the liquid part or liquid component what has been participating into the immune response. So it could be antibody, it could be complement, it could be all the liquid components which are being secreted from the cell, and it is a vital component of the adaptive immune response. So it primarily defends against the extracellular pathogens such as bacteria, viruses and toxin by producing the antibody that circulate into the body fluids, and that's why historically it has been considered or referred as humors. What are the component of the immune response? So you have the B lymphocytes, you have the plasma cells, you have a memory cell. So the B cells develop into the bone marrow and express using the B cell receptor on their surface for each specific particular antigens. Upon encountering the specific antigen, B cell becomes activated and differentiated into the plasma cells and the memory cells. Plasma cells are the effector cells that secrete the large quantity of antibodies specific to the encountered antigens, whereas the memory B cells are long-lived cells that remember the specific antigens, enabling a rapid and robust immune response upon the re-exposure. Then we have the antibodies, right? So antibodies are Y-shaped protein produced by the plasma cell that specifically bind to the antigens. So we will discuss about the structure of antibodies in our subsequent lectures, right? So that time you will understand more about these antibodies. There are 5 main classes. You have the IgG, IgA, IgM, IgE and IgD, and each with a distinct role into the immune system, right? Then we also have the helper T cells or the CD4 T positive cells that assist in the B cell activation by recognizing the antigen presenting B cell and secreting the cytokines that produce the B cell proliferation and the differentiations. Then we also have the complement system. So all these are part of the humoral response. A group of proteins that are then activated by antibody bound to antigen enhance the ability to clear the microbe and the damaged cells, promote the inflammations and attack the pathogens plasma membranes. Then we also have the cellular system. So cell mediated immunity is a crucial component of the adaptive immunity system that defends the body against the intracellular pathogens such as viruses, certain bacteria and the cancer cells. Remember, the humoral response actually protects the body from the external or extracellular pathogens such as bacteria, viruses, fungus and so on, whereas the cell mediated immunity primarily protect from the intracellular pathogens such as viruses, bacteria and the cancer cells. Unlike humoral immunity, which relies on the antibodies, the cell-mediated immunity involves the activation of specific cells to identify and eliminate the infected or the abnormal cells. Number one is the T lymphocytes or the T cells. And so T lymphocytes could be of two types, cytotoxic T cells or CD8 positive cells. These cells recognize and directly kills the infected or the cancerous cells, presenting the cell specific antigen bound to the MHc class 1 molecules. Then we have the helper T cells or the CD4 positive cells. They assist other immune cells by releasing the cytokine that enhance the immune response. Then we have the macrophages. So these phagocytic cells ingest and destroy the pathogens and present the antigen to the anti cells and bridging the innate and the adaptive immunity. Remember, all these cells are part of the innate immune response, but when they encounter a pathogen, they get trained to elicit an immune response, and then they become also a part of the adaptive immune response. Then we have the natural killer cells, right? So part of the immune cells, NK cells, can recognize and kill the infected or transformed cells without the prior sensitization, often detecting the cell with the down-regulated MHc class 1 molecules. Then we have cytokines. These signaling molecules such as interferon gamma and TNF alpha are produced by the T cells and the other immune cells to regulate and amplify the immune response. What is the function of the cell-mediated immune response? It is targeted and destroys the cell infected with the viruses or certain bacteria that resides within the host cells, for example, the TB, right, or Mycobacterium tuberculosis, right. So Mycobacterium tuberculosis is an intracellular pathogen. So it is an intracellular pathogen. So what the body resides, it is actually going to destroy the whole cell, right. Then it identifies and eliminates the cancer cells presenting the abnormal antigens, and then it recognizes and attacks the transplanted tissue or the organ perceived as a foreign, right. So cell-mediated immune response is working against the big objects like or the intracellular pathogens. Then we have different types of immune responses, right. So we have the immune responses when the pathogens are being infected. So pathogens are the microbes that cause the disease, including the viruses, bacteria, fungi, and the parasites. Pathogenesis refers to the process through which these organisms invade the body and cause the illness. The immune system identifies the pathogen by detecting the unique molecular pattern that are not found in human cells, right? So you are actually going to have a unique pattern what is being present onto the outer surface of these pathogens, right? And that pattern is not present in human system, right? So what will be the first line of defense? There will be a physical and the chemical barriers. So skin and the mucous membrane, which is actually going to work as a physical barrier, and it is going to block the entry. Then we also have the acidic environment in the stomach, vagina, and sweat, and that prevents the microbial growth. And when these barriers are compromised, such as you are going to have a cut, there will be a burn, there will be an insect bite. So when the insect bites, it actually injects directly the particular pathogen into your skin, right? So it doesn't get blocked by the all these kind of barriers. The risk of the infection increases significantly. Then we have the role of the microenvironment. So body's location and context influence the immune responses. Some regions, for example, the central nervous systems are protected from the full immunity to prevent the damage. Then we have a tolerance. Tolerance occurs in the digestive tract for harmless or the beneficial microbes. And the same microbe may trigger a strong immune response if it enters the bloodstream instead, right? So basically there are places which are being tolerated, which are being not be immunologically that active. For example, you have a digestive tract, you have the fallopian tubes, and you have the that tract of vagina and all that. That place is immunologically not very highly active. So because of that, it actually allows the proliferation of harmless and the beneficial microbes, so that you can actually have the gut microbe flora and all those kind of things, because all these microbes are actually going to help you in terms of producing the different types of vitamins. It is also help you in digesting the food material and so on. So recognizing triggers the immune response. The immune system is activated only after pathogen breach the physical barriers and the chemical barriers. So host cell uses the recognized molecule to detect the whole pathogens, pathogenic fragments like the or the pathogen secreted products, right? So these are the three categories which are actually going to be recognized by the immune system as the non-self, right? Either it is going to recognize the whole pathogen like the whole bacterial cell. It could actually be able to recognize the pathogen fragment like the broken bacteria, or it can also be able to from the pathogen secreted product. Then binding these molecules start a signal cascade leading to the destruction of the invaders. Then we have the intracellular pathogens such as viruses or some of the specific bacteria or the cancer cells. Like, so viruses are live and replicate inside the host cells, hiding from many immune defenses, right? So cytotoxic T lymphocytes detect the changes in the infected cell and kill them, right? And the intracellular receptors alert the immune cells to the presence of viruses inside the cytoplasm. For example, HIV targets the helper T cells and weakening the immune system and leading to the immunodeficiency. Then we have the extracellular pathogens such as bacteria, fungi, some parasites. So found outside the cell, these are targeted by the B cell and the antibodies. And antibody binds to the pathogens, marking them for the destruction or the neutralizing them directly. Antibodies can reach spaces where the immune cells may not be able to go. Now let's talk about some of the immune responses. So the first immune response is the phagocytosis, right? So phagocytosis is basically a process which is called as eating, right? So eating by the cells, right? So despite the strong defense of our protective epithelial cells, some pathogens have evolved strategy to penetrate these defenses, and epithelia may be disrupted by wounds, abrasions, insect bite, and that may transmit the pathogen. Once the pathogen penetrates through the epithelial barrier into the tissue space of the body, an array of cellular membrane receptors and a soluble protein that recognize the microbial component play an essential role of detecting the pathogen and triggering the effective defense against it. So phagocytic cells make up the next line of defense against the pathogen and have penetrated the epithelial cell barriers. So we have the macrophages, neutrophils, dendritic cells in tissues, and the monocytes into the blood are main cell type that carry out the phagocytosis, that is the cellular uptake of the particle material such as bacteria, a key mechanism for eliminating the pathogen. So phagocytosis means cellular uptake or the eating of a particular material, right? How it happens? So what is the mechanism of phagocytosis? So through the cell surface receptor, the cell recognizes the microbes such as bacteria, extend their plasma membrane to engulf them and internalize them in a phagosome, right? So this is what exactly happens. The bacteria become attached to the membrane invading, and so when the bacteria actually touches the membrane, right, it actually induces a series of signaling cascade, and because of that the pseudopodia comes out from this particular cell, and slowly these pseudopodia are actually going to be fused, and as a result of this the bacteria is going to be encircled into a membrane vesicle. This membrane vesicle is called as the phagosome. Phagosome fuse with the lysosomes, right? So lysosome then fuse with the phagosomes, delivering the agent that kill and degrade the microbes. Remember that the lysosomes are actually having the hydrolytic enzymes. They are actually going to have the low pH, and they are actually going to destroy all the bacterial material, right? Neutrophils are second major type of phagocyte that usually recruit to the site of infections, and finally the bacteria is killed and digested by the lysosomal enzyme, and digested products are released from the cell, right? Some of these digested products such as the small peptides are also going to be expressed along with the MHc molecule, and that's how they will be actually going to give the signal to the neighboring cells, like they are going to give the signal to the T cells and B cells to elicit more pronounced immune response. So basically the phagocytosis is to eliminate the pathogens and to also put an alarm to the immune system into the body that okay, there is a pathogen of this particular type of peptide or this particular type of antigenic characters, and let's induce an immune response against this particular type of antigen. So the second part is the antigen presentation, right? And for the antigen presentations, we have to understand about the major histocompatibility system, or the MHCs. So the MHC complex is a collection of a gene arrayed within a long continuous stretch of DNA on a chromosome 6 in the human and a chromosome 17 in the mice. The MHC in human called HLA, or the human leukocyte antigens. There are three different types of MHC molecules. You have class 1 MHC molecules, you have a class 2 MHC molecule, and you have class 3 MHC molecule. So in a class 1 MHC molecules, which are encoding the glycoprotein expressed on the surface of nearly all nucleated cells, and presentation of a peptide antigen to the T lymphocytes. Okay, then you have a class 2 MHC molecule which are encoding the glycoprotein expressed primarily onto the antigen presenting cells, which are called as macrophages, dendritic cell, and the B cells, and presentation of peptide antigen to the T helper cells. Then we have a class 3 MHC molecules which are encoded with the in the other kinds of products like secreted protein that have immune functions, including the component of the complement system and to the molecule involved into the inflammation. Then we have different types of antigens. You have the endogenous antigens, you have the exogenous antigens. So endogenous antigens, that is antigens which are processed within the cytoplasm, that could be viruses or bacterial infected cells. So these are displayed on the surface of the class 1 MHC molecules, and the pathway for the processing is been called as the cytosolic pathway. Whereas for the exogenous antigens, these antigens are processed by the endocytosis, and it could be any foreign substances. They are displayed onto the surface of the class 2 molecules. The pathway for the processing and presentation of exogenous antigen is called as the endocytic pathway. Now first talk about the endogenous processing pathway. So large endogenous antigen is either viral or bacterial infected proteins are digested into the small fragment by the ubiquitin mediated proteasome. The abnormal protein that are digested into small peptide fragment with the size of 8 to 10 amino acids. Then the digested peptide fragment transported from the cytosol to the RER mediated by a dimeric protein on the surface is called as the TAP. So it is, and then alpha chain and beta chain is synthesized into the polysomes of the RER, and a type of molecular chaperone called calnexin present in the RER aids in folding of alpha chain and releasing into the binding of beta 2 microglobulins. And the another molecule which is called as tapacine associated with the folded class 1 molecule and bind with the peptide and express onto the surface of antigen presenting cells. Then we also have the exogenous processing. So antigen presenting cells can internalize antigen by the phagocytosis, endocytosis, or both, and then the macrophage internalize antigen by both the processes. Once the antigen is internalized, it is degraded into the peptide within the compartment of the endocytic processing pathway. Internalized antigen takes 1 to 3 hours to transfer the endocytic pathway and appear it at the cell surface in the form of the peptide class 2 MHC molecules. The endocytic pathway appear to involve three consecutive acidic compartments: the early endosomes, late endosomes, and the lysosomes, right? Internalized antigen move from early to late endosome and finally to lysosomes, encountering the hydrolytic enzyme and a lower pH in each compartment. Within the compartment of each endocytic pathway, the antigen is degraded into the oligopeptide of 13 to 18 residues, which then bind to the class 2 molecules. When the class 2 molecules are synthesized within the RER, three parts of the class 2 molecule associate with a pre-assembled trimer of a protein which is called as the CD74. This trimeric protein interacts with the peptide binding cleft onto the class 2 molecule, preventing any endogenously derived peptide from binding into the cleft while the class 2 molecule is within the RER. The invariant chain also appear to be involved into the folding of the class 2 MHC chain. Their exit from the RER and subsequent routing of the class 2 molecule into the endocytic processing pathway. The invariant chain is digested and leaving the peptides called as CLIP, or the class 2 associated invariant chain. A non-classical class 2 molecule called HLA-DM is required to catalyze the exchange of CLIP with the antigenic peptides, and then HLA duo also called to dissociate the HLA-DM and finally dissociate with the antigenic peptide, and the class 2 MHc with the peptide activity activates the T helper cells. So this is what exactly it shows into the exogenous processing pathway, where first the antigen is actually going to be bind by the receptor what is present onto the cell surface, then it is actually going to be internalized into the vesicles. Then these vesicles will pass through different steps where they are first going to be into the early endosomes, then they will be into the late lysosomes, and then it is actually going to be where all the antigen is going to be digested, right? And then it is going to be bound with the MHc class 2 molecules, and then ultimately the MHc class 2 molecule containing peptide is going to be expressed along with the MHc class 2 onto the cell surface of the antigen presenting. So this is the major steps what is happening into the endogenous antigen versus the exogenous antigen. Then we also have another aspect which is called as graft rejections and the MHCs, because MHCs are, we said that we have the two important different types of MHCs, MHc class 1 and class 2, and class 1 is present on the all cells, whereas class 2 is only present onto the antigen presenting cells. So class 1 is actually a representation of or the specific to a particular organism, and the class 1 molecules are actually should be matching with the another person with the class 1 molecule, and that's why that is the major basis of the graft rejections, major basis of the transplant, right? If that is not matching, then it is actually going to do the graft rejections. So what are the different types of transplant? So the degree and type of immune response to a transplant varies with the type and source of the grafted tissue. The following terms denote the different types of transplants. You have the autograft, you have the isografts, and you have the allograft. So autograft is the self tissue transferred from one body side to another in the same individual. So autograft means you are taking out skin from the thigh and putting the skin onto the face. So it is actually the same tissue, but the location is being different. Examples like transferring the healthy skin to a burned area in the burned patient, and usually healthy vessels to replace the blocked coronary artery. Then we have the isograft. The isograft is transferred between the genetically identical individuals. This occurs in an inbred strain of mice or identical human twins when the donor and recipients are the synergistic. Then we have the allograft. Allograft is a tissue transferred between the genetically different member of the same species. For example, one person is taking one organ from the different person or from his family member, right? So in mice, this means transferring tissue from one strain to another, and in humans, this occurs in the transplant in which the donor and recipients are not genetically identical. Then we also have the xenograft. So xenograft means the tissue derived from the different species. For example, you are taking a graft of the heart from the pig, right? So you're going to have a pig heart into the humans. So because of a significant shortage of donated organs, raising animals for a specific purpose of serving as a organ donor for human is under serious consideration. So autograft and isograft are usually accepted owing to the genetic identity between the donor and recipient. Because an allograft is genetically dissimilar to the host and therefore express unique antigen, it is often recognized as a foreign by the immune system and therefore rejected. Obviously, xenograft exhibit the greater genetic and antigenic specificity, enabling a vigorous graft rejection, right? Because xenograft means you are taking you have changed the species also, right? So in that case, the immune response will be more robust compared to the allograft. Now, what will be the role of the MHCs? So the most intense graft rejection occurs due to a difference between the donor and recipient in terms of the ABO blood group and the MHc antigens. So the blood group antigens are expressed onto the RBCs. You know that you have A antigen, you have B antigens, and then you have, so A and B antigen combinations gives the four blood group, right? You have A blood group, B blood group, AB blood group, and if the both Na, B are not present, then it becomes a O blood group, right? So epithelial cells and endothelial cells requiring the donor and recipient first to be screened for the ABO blood group compatibility, which means if you want to do a, if you want to check whether the two person can receive can take up the, the organ from the other person or not, the first thing is their blood group should be identical, right? And then if the recipient carries the antibodies to any of the antigen, the transplanted tissue will induce the rapid antibody mediated lysis of the incompatible donor cells, right? So for this reason, most transplants are conducted between the individual with a matching ABO blood group. Next, the MHc compatibility. So MHc compatibility between potential donor and recipient is determined, which means you are going to check whether the MHc-I what is present onto the donor and the MHc-I what is present onto the recipient is same or different. If they are different, then it is actually going to recognize that as a foreign antigen, and then it is actually going to elicit the immune response. So if these two are same or similar, right, then there is a possibility that you can take the organ from the one person and put it into the another person. The first choice is usually patterned for the first order sibling with at least a partial MHc match, followed by other family member and even the friends. Given our current success with the immunosuppression and the immunes into the recipient prior to the translation, the importance of careful cross-matching was shown in seminal 1969 study, where the 80% of the kidney transplantation with a positive cross-match experienced almost immediate graft rejection, while only 5% patients with a negative cross-match had this outcome. The MHc makeup of donor and host is not the sole factor determining the tissue acceptance. Even when the MHc antigens are identical, the transplanted tissue can be rejected because of the difference at the various other loci, for example, the minor histocompatibility locus. The major histocompatibility antigens are recognized directly by the T helper cells and the T cytotoxic cell, a phenomena that becomes the yellow reactivity. In contrast, the minor histocompatibility antigens are recognized only when the peptide fragments are presented in the context of the self-MHc molecules. So rejections based on only the minor histocompatibility difference is usually less vigorous and can still be lead to the graft rejections. Therefore, even in the class of HLA identical matches, some degree of immune suppression is usually still required. Now, the second response is called as the inflammation, right? You might have experienced the inflammation, right? For example, when there will be a bite of honeybee, for example, so that portion becomes red and it becomes swollen, right? So that is been because there will be an inflammatory reaction. So when the outer barrier of the innate immunity, for example, the skin and the epithelial layer are damaged, and the resulting innate response to infection or the tissue injury can produce a complex cascade of events which are called as inflammatory responses. Inflammation may be acute, which means the short term, or inflammation could be chronic, for example, the long term, contributing to the conditions such as arthritis, inflammatory bowel disease, cardiovascular diseases, and type 2 diabetes. The hallmark of a localized inflammatory response was first described by the Roman physicians in the 1st century AD as robust tumor at low redness and the swelling with the heat and the pain. An additional mark of inflammation added in the 2nd century by the physician is the loss of function, which is, and today we know that these symptoms reflect an increase in vascular diameter, which is the vasodilation, resulting into the rise of blood volume into the area. Higher blood volume heat the tissue and cause it to the redden, right? Vascular permeability also increases, leading to the leakage of fluid from the blood vessels, resulting in accumulation of fluid, which is edema, right? So it becomes swollen, right? You might have seen that it becomes swollen, and then you have the different types of the skin becomes slightly, loose, right? Within an hour, the leukocytes also enter the site from the local blood vessels. These hallmark feature of inflammatory response reflect the activation of the resident tissue cells, for example, the macrophages, mast cells, and dendritic cells, to release the chemokines, cytokines, and other soluble mediator into the vicinity of the infection or the wound. Recruited leukocytes are activated to the phagocytosed bacteria and debris to amplify the response by producing the additional mediators. Resolution of this acute inflammatory response include the clearance of invading pathogens, dead cells and damaged tissues, the activation of the systemic acute phase response and additional physiological responses including the initiation of the wound healing and the induction of the adaptive immune response. However, if the infection or tissue damage is not resolved, it can lead to the chronic inflammatory state that can cause more local tissue damage and potentially have a systemic consequences of the infected individual, right? So this is what exactly is happened, right? Suppose you got, the prick of, the thorn actually. So if you got a prick of thorn, it is actually going to get inserted, right? And that is actually good enough to inject some of the bacteria, right? So in response to bacteria, right? The first thing will happen is if there will be a tissue damage and bacteria cause the resident cells to release the chemoattractant, that will also cause the local increase in blood vessels and capillary permeability. So it is actually going, what will happen is this area is going to be swelled, right? It is going to swell because there will be a vasodilation, and because of the vasodilation there will be a more supply of blood, and that's how it becomes red in color, right? And then the second thing is it is actually going to invite the all sort of phagocytic cells. It is going to bring the neutrophils, it is going to bring all those cells, right? So that there should be a robust immune response could be generated, right? And that's how it is actually going to help in terms of developing the, it gives the immune system to protect themselves against the different types of infectious organisms. Because anything which actually going to break the physical barrier will be alarming for the body, and that's how the body has made a system where it is actually going to start the cascade of the inflammatory reactions, where different types of phagocytic cells are going to come at the site. It will actually make a protection layer against that particular infectious organisms, and it is also going to increase the blood vessel and the blood volume, so that more and more immune cells could actually become at the site of the infection, and that's how it is actually going to cause the inflammation. So bacteria enter through a wound, right? Initially innate immune response, right, including the phagocytosis by the activation of resident cells such as macrophages and dendritic cell. Recognizing the bacteria by the soluble and cellular pattern recognition, molecule initiates an inflammatory response that recruits the antimicrobial substance and phagocytes to the site of infections. So, so far what we have discussed in this particular lecture, we have discussed about the importance of the study of the immune system, right, and the development of a branch of the science which is called as immunology. And we have also studied, we have also understood why there is a need to study the immune system and why, how it actually helps you in terms of developing the different types of vaccines. It also helps, and what we have also discussed about the different components of the immune system. So we discussed about the innate immunity response, we discussed about the adaptive immune response, and in terms of the executory molecules, we have also discussed about the humoral response or humoral immunity. We have also discussed about the cell-mediated immunity. So within the humoral immunity, we discussed about the antibodies and other kinds of molecules which are participating into the immune reactions. And within the cell-mediated immunity, we have discussed about the the B cells, T cells, and macrophages, and all those kind of cells which are involved into the to the responding to the different types of foreign antigens, and how they are actually eliciting the immune responses. As far as the immune response is concerned, we have also discussed about the phagocytosis, we have also discussed about the inflammations. And very briefly, we have also discussed about the MHc molecules, we have also discussed about its relevance in terms of determining the tissue compatibility between the donor and the acceptors. So with this brief discussion about the basics of the immune system, I would like to conclude my today's lecture. In a subsequent lecture, we are going to discuss some more aspects of the molecular immunology. Thank you.